Everyone. Well, thanks everyone for coming. I hope you guys can hear me well. I have a little bit different accent than Jeff. It's a little bit worse because I'm originally from Brazil, but I've been living in the US for the last 10 years. I've been with the company. I work for Varietal Biosciences, which is a sister company for Sumitomo Chemical that probably are familiar here in Australia. But prior to that, I was at Cornell University for 4 years, working specifically with the plant growth regulators. Today, my main talk here is going to be around flower formation, but we can discuss a lot of things in the field about PGRs in general, because that's what I've been working with. Nick asked me to specifically talk about the work I've done during my thesis and during the 4 years I was at Cornell. And the topic was brought up by Cornell in New York, or because of the issues we have with the Honeycrisp, as you heard from Jeff, tends to be biennial. But what I'm going to show you here, it can be applicable to any other biennial variety you have. Okay? I hope you guys are going to learn something from me. Just let me know if you don't understand something. I want to make things clear. Okay? So, apple formation, flower bud development, and return bloom. So before I go, the first 2 slides is just to set the stage for the field discussion. So plant growth regulators, what they are. So quite often, plant growth regulators are set more like an investment. It's a little bit different than fungicides and insecticides, but you apply plant growth regulators because you want a high fruit quality, you want to reduce your labor cost. They are there to help, to increase your profitability. So PGRs are often considered, they are naturally occurring, which means any plant hormone that's being produced by the plant can be considered a PGR, or it could be a synthetic organic substance. A plant, for instance, you are familiar with Regalis, Harvista, Retain, Silex. Silex has a cytokinin, but the cytokine is a synthetic cytokine. All those are PGRs. However, if you know a GA3 product, ProGib, for instance, is considered a plant hormone because the tree itself produces GA3. So those are the small differences. Usually we talk about parts per million, and I know growers hate parts per million. They want to know the amount of product you want to apply in the field, exactly what you want to do. But a PGR is often, because we're talking about things that can modify the development of the tree or the quality of the fruit, usually we talk about parts per million. Today, when we look in the literature, we have around 9 main plant hormones that's being discovered, and we know what they do in the plant. But the main ones that are very clear for us is the gibberellins, auxin, cytokinase, abscisic acid, and ethylene. And I think most of you may be familiar with those. I usually like to say that the 3 first ones here, the gibberellins, auxin, cytokinase, they are related, usually they control plant growth. They induce plant growth because of their role on cell division. And they're often, if you measure the level of these plant hormones, they're often higher early in the season as the tree is growing, as the fruit is growing, etc. Those 2 other hormones, abscisic acid and ethylene, they're more like an aging hormones. They are responsible more for maturation, for senescing. When the flower is senescing, it's producing more ethylene. When the fruit is maturing, it's producing more ethylene. Most of you have used Harvista Retain to reduce the ethylene levels, right, to prolong the life of the fruit. So this is a little bit the difference. When we talk about apples, apples are very unique. Apples are very unique because we actually have PGRs for any time of the season. I'm not saying that you guys have PGR for everything, but when I combine globally all the PGRs that are being used, we have PGRs that can be used early in the season for branching, for stem elongation of the flower. We can use PGRs to control the growth, to reduce or to increase the growth, to increase fruit set. We have PGRs for thinning. Most of the thinners are considered PGRs, for fruit color, for harvest management, post-harvest management, all the way to defoliation. We still don't have a product that could efficiently put the tree into dormancy. There's a lot of questions. When I was visiting Western Australia, A lot of questions about how can we use Abscissicast to put the tree into dormancy to help. We are doing some work on that side, but right now there's nothing there. But you can see the amount of PGRs. So we can discuss. If you guys have any questions, I've been working, my role in the company is to give technical support to growers. We can discuss that more in the field. So flower and fruit development, nothing is new. I'm gonna take you back a little bit to school and just to emphasize the importance of thinning, basically. You're gonna see at the end of my presentation. But everybody knows that the fruit that you're gonna harvest next year has been produced right now. The tree actually is already start forming the flowers and the fruit that you're gonna harvest next year. The main sites of fruiting buds are the spore buds and also in 1 year old, which you call burst shoot, on the terminal of the burst shoot, or even on the axillary buds. Often we are just concerned, we want a flower and fruit on these spore buds on the terminal bud. These flowers here, usually they are the poor quality. When is this process is happening then? So these are common time of flower development on spur buds. For instance, you have from mid-late spring, which is around now, all the way to spring next year. So it takes almost more than a year for one bud to form a flower. I like to separate those stages into 4. So first you're gonna have the flower induction. So that's a transition from vegetative to flower. When I say flower, actually a bud can be vegetative or a mixed bud. That means it's a flower, but also has a vegetative that is gonna keep your crop for the next years. So it's always a mixed flower, but we often say flower bud. So that timing is the tree telling the bud, preparing the bud to be a flower or to be vegetative. We don't see that stage, it's the transition. The first stage we can see is the flower initiation, which is if you bring this bud here, right now, for instance, if you start bringing the buds to the lab, you dissect the scales, you open and look at the meristem. If there is a dome, it means that bud's gonna be a flower. It's the flower initiation. I can tell you, I can separate 30% of our buds is gonna be flower or it's gonna be vegetative. And that happens for some varieties you see that may be happening right now. Then you have the flower differentiation, and that's when, once you know it's a flower, then you're gonna start forming the king flower, the lateral flower, and then you have the sepals, the petals. All those processes happens throughout the fall, through the winter. And then the last 2 portions that is gonna be formed is the pollen and the ovule. Those are gonna take place from the green tip all the way to bloom. So that's the stage. So when you look here, while you're taking care of this year crop, you are also taking care of the next year crop. Because something happened here, you're gonna end up with some stress happened somewhere here, you're gonna affect the quality of your bud next year, okay? So just to make it clear. Jeff talked about when they do the bud analysis to see which one is flower, quite often you're gonna be focused on this stage here. And I'll show you that in a bit. So this is the study I've done from 2014 to 2018. And actually I continued when I moved to New York, where I was looking at the timing of flower initiation. So it's really hard to see this. You can see it under a dissecting microscope, but you have to have good eyes to be able to see the doming. I want to tell growers when that is happening, because if you want to do something, you have to do before that, because once it's a flower, it's there. So this is the bud at stage, the percentage of buds at this stage here. If I look at Gala and Fuji, the first thing I can see is that Gala usually initiates flower a little bit later than Fuji. The peak often is 85 to 95 days after bloom. And Fuji is a little bit earlier. In Fuji, tends to be biennial. If I compare the 3 sites of fruiting units, the spur bud, the terminal, and the axillary bud, the spur bud, which is in red here, it always forms a flower before. It's the first one to have a flower formed. And then initiate, in this case. And then some years you can have this terminal bud, and some years you can have the axillary buds. In this case, we had the axillary bud first. It's going to depend on the growth of the shoot, because the terminal bud is going to only be formed once the growth stops. But the axillary flowers can start forming. When we saw, okay, Fuji is early, how about Honeycrisp? That's why we start doing some more work on Honeycrisp. I know you don't have Honeycrisp here, but think about your biennial varieties. So Honeycrisp is way early, and just as Jeff mentioned, we have huge issues with return bloom the variety, because the peak is about 45, 55 days after full bloom. I'm telling you that I'm telling you that like maybe 80% of the buds are going to be formed at this stage. It doesn't mean that you're not going to have some flower formation later. We will see in a bit. But basically, these are the timing. So we decided to look into other varieties just to confirm. Maybe the varieties that tend to be biennial, they formed the flower, they initiated the flowers earlier. So these are days after full bloom. I know plants don't count days after bloom, but that's the easiest way for us to explain you guys when is that timing happening. June, July, August, forget about because it's not the same here. Just pay attention days after full bloom. So you have the flower induction, which is the preparation of the tree. And then you have the flower initiation from different varieties, and then you have the differentiation. As you can see here, Honeycrisp, Minesca is the sweet tango, apple, Macau, Fuji, Gold. All these varieties tend to be biennial. And they initiate flowering earlier compared to our Gala here, or even the US, they're way later. That's why we don't see a lot of problems with flower initiation. Minesca, I'm not gonna get into this topic here, but if you overcrop this tree, it's gonna turn biennial. The only thing it doesn't turn biennial is because it self-thins and has way less seed numbers within the fruit. But we are the experiment overcropping and putting more bees, more pollen, and it tends also to be biennial. It's just as early as Honeycrisp. So our conclusion was that varieties widely differ in the time of flower initiation or flower induction. Varieties that are prone to biennial berry initiate flower earlier than the annual crop varieties. Now, when we talk about differentiation, I don't know if this is going to be so applicable for you as it was in Western Australia, in WA, where they have poor winter chilling. They have poor chilling accumulation there during the winter time. And that can explain. So during my thesis, when I was doing my PhD, I separate into different stages and want to know, this is the flat when it's the stage 0 is vegetative, the initiation, which has the domain, and then you have the king start being formed, the laterals, flowers being formed, and then you have the sepals being formed, and then you have the anthers. So all this is happening throughout the season, the anthers, and then the pistil that's going to receive the pollen until you have a complete formed flower. So I looked into the conditions, how that performed in Brazil for 2 years and how it performs in the US. I did the US only for a year, and I picked the Fuji variety. If you look here, these are days after full bloom, and what I have in red is in the US, and the other two colors, black and gray, are in Brazil. And here I have all these stages. As you go longer, so stage 1, which is this doming, there is no difference between the two countries. The time initiation happens at the same time. We count 45 days, 65 days after full bloom, depending on the variety. What differentiates us from the other, for the US, in Brazil from the US, is because once I know this time, from here to here in Brazil is very quick. Very quick. It's quicker compared to the US. The US, it takes longer for the bud to be formed. We look at the temperature. The red is the US. You see the gray and the dark. This is the average temperature. It's flatter because we don't have good chilling unit. We have poor winter in Brazil. So, and it's really warm. Probably the temperature has an effect on the differentiation, not on the initiation, but it probably make that buds to form faster. And that's a problem for us because we don't go in, the flower is already there, and we don't go into dormancy. So the tree keeps metabolizing and keeps using energy, and that wastes energy, wastes uses carbohydrate from the tree, and you don't have enough accumulation there. And that's why when we compare the size of a flower in Brazil with the US, it's way smaller. So that affects the quality. I don't know if you grow pears, but pears, they differentiate way earlier than apples. Like for instance, even considering Brazil, it's quicker, but around, I can't remember the total days, but around harvest, you already had the whole cluster formed. And that's one of the reasons we can grow pears twice a year in Brazil. And where we are close to the equator, we have a low chilling variety that requires low chilling. We can have 2 crops. They cut the irrigation and then continue the crop, because that forms very early. We are not good producers of pears because of the bud abortion in the southern region where we grow, but that explains a lot. When I talk about initiation, but that's what I was looking into under the microscope to tell you when that happens, you guys probably, especially in the US and some other countries as well, they do the bud analysis to see what is the percentage of bud that is going to be flower or vegetative. And if you do this longitudinal cut, as Jeff was telling us before, you can tell which one is going to be flower, which one is going to be vegetative. And that's often, if you do this around the harvest time or after harvest, you can clearly see which one is going to be flower. That one has a dye, just for you to see better, but you can see the flowers here compared to a vegetative bud, which is flat. You have only primordia. So this is very easy to do in the field. This is what we call the differentiation time. What happens when you do summer pruning? I explained to you that maybe 80% of the buds are going to be formed at critical time in there. That's 65 days, Fuji, for instance. So that's where you should be paying attention. If you want to do anything to improve return bloom next year, when we should be doing? During the flowering induction time, not during the flower initiation time. I think on this graph, maybe it shows better. This is the time where you should be doing all the strategies, most of the strategies to increase your return bloom for next year, okay? So when you do summer pruning, or use hedgers, or you have some regrowth, right? And every country has a different time to do that. When you have those regrowth there, and if it's not too strong, and if the tree is not overcropped, because if the tree is overcropped, you're not going to have a terminal bud, a flower bud there. And this is what happens. You have some regrowth there. The bud goes. That's the winter time. In the US, winter time, we should be already, if I collect buds, regular buds, I would have already differentiation. Most of the king, the letter be formed. But because of that regrowth, I do have initiation there. You see the doming there of the meristem? I have flower formation later because of that regrowth. So flower induction apple is not controlled by temperature day length. It's not about that. It's about the balance of the plant hormones. There's other factors affecting the flower induction. A lot of growers do, I've seen around the world growers doing regalis after to hold the growth and then put etrel. And I'll talk a little bit about the plant hormones. Ethefone is ethylene, producing ethylene. Ethylene actually helps just to hold back and form a flower on that bud there. Now, you may ask me, what is so if you have a late flowering or a late bud, a flowering bud, what is the quality of this fruit? Because you won't have much time from, that's January, and then bloom in New York, for instance, is going to be May. It's a short time. I don't know. I've never tracked back to see the quality of the bud, quality of the fruit of this bud in particular. But it does happen later in the season. So when you talk about the indirect factors, I won't go through all of them, but there's 2 main ones I want to point out here. Okay? So crop load and thinning. And everybody knows, if you want to get good return bloom, you have to thin. But thin sooner. For those varieties that you have problems with biennial berry, you got to do thinning early because the fruit carries the seeds carry gibberella. And gibberellins, which is the plant hormone that can suppress flower formation. If you have too much apples, too much seeds, too much gibberellins, and that's going to affect your return bloom for next year. So it's important to do early thinning, and I'll show you in a bit. And then there are other things that can affect negatively or positively your return bloom. And then also the other topic that a lot of people ask is about the use of plant regulators. Gibberellins to reduce your return bloom for next year. Or you can use ethylene to promote return bloom. So, yeah. So yeah, we have actually a label for the use of GA47 for increase the length of the stems. According to our studies, at that time when you spray, it's from tied cluster to early pink bud. There is no effect on return bloom. It's very, very early, according to that time. We can talk more about that in the field in regards rates and timing, exactly that.
That was my question too. It's about when's the timing on that?
Yeah, we can discuss that in the field. So when you talk about plant hormones in the plant, so ethylene, abscisic acid, cytokinase are the proven plant hormones that can induce return bloom. Auxins and gibberellins, they reduce the return bloom. The others to increase the return bloom, sorry. Quite often you hear about sprays, summer sprays, and we'll talk in the next slide. But talk about just the thinning part. And this is a very simple trial to show the timing of thinning versus the return bloom. So what we did in this study, Honeycrisp, but hypothetically you can think about any other variety that tends to be biennial because you're going to see the same example on Fuji. We had trees where we didn't thin anything off. The other trees, we went at bloom and left the king flower. Okay, every single cluster thinned down to one flower per cluster. And then we went there at balloon stage to thin down. And the other treatment, we waited until 10, 12 millimeters fruitlet size, and then we thinned down every single cluster to one fruit per cluster. Around this time, we adjust the crop load of this treatment as well, because our target for this tree was about 80 to 90 fruit per tree. And we decided, let's leave more. Let's see if the tree can handle more. So this is way more fruit than we needed. So that's the year we did the hand thinning. Crop load is the same because crop load can affect the return bloom. But we want to see, we have to adjust the crop load the same. When I look at my fruit size, of course, if I leave my king flower there, I have bigger size than I do if I do a late thinning there. Sorry, a late thinning there. We did another study on this variety that tends to be big, large fruit. In this variety, not all varieties are like this. If you leave the smallest flower on the tree, on the cluster, you reduce the fruit size. Instead of getting 256, you can down to 210. Just leaving the smallest flower within the cluster. Some other varieties, doesn't matter what flower you leave, you can increase the size. We can talk about other strategies, how some of the growers we played in New York to do for the thinning for that. But size is not what we are looking here. Return bloom. If you do your blossom thinning, you get a better return bloom next year, and then we counted the number of fruit per tree. Of course, we could have left less fruit per tree, but it's still, compared to my 10, 12 millimeters fruit size, I had better return bloom when I do the blossom thinning. Same thing, Fuji, same exercise. Again, size is better, and I had way better return bloom than when I did the 10, 12 millimeters fruit size. These trees, we really overcrop because it was around 100 fruit per tree. We left way more. Sometimes if you do early thinning, you can allow the tree to carry more fruit. So if you do more earlier. Now, how can you get the number down at bloom? That's the magical question, because we don't have any silver bullet recipe or any product that could be a perfect thinning. But the thing is, you guys have to be more aggressive early, especially for this variety. You have to do that early thinning. Now, when you talk about using now Etefone or NAA as in a spray program during the season. In the past, at least in the US, and Jeff here can confirm, a lot of growers were doing the late sprays. You may ask me, NAA is auxin. You just told me that auxins can reduce return bloom. But early in the season here, the tree is producing a lot of auxins. And if you put more auxins, that's going to trigger ethylene production. And that's what NAA does. It triggers the ethylene production. That's why you can get thinning early in the season. If you put the auxins later in the season, you can hold the fruit on the tree, right? Because there's not much auxin. You're supplementing the auxin later in the season. But that's why NAA is used. So usually they do summer sprays. I've done, I don't know, 10 years of trials. Since I was at my PhD, I never was consistent with the return bloom. I worked a lot with Fuji, was never consistent with that. So after seeing that its initiation is way earlier on those varieties that tend to be biennial, we're not doing right. We should be spraying earlier than that. We should be, thinning should be the first priority here. Instead of spending money on PGR, I sell PGRs, I work for a company that sells PGR, but thinning should be your first priority. And I mean early thinning, more aggressive here. But how about supplementing the plants with PGRs here earlier, not that late? So we did a study at Cornell. We've been working the last 4 years, 3 years, I think, I don't know. But we decided to use Etral, ACC. Probably a lot of you have heard about the Exceed, the new product that's coming in the market. Also generates ethylene. So we decide, the good thing is because there is no residue, and for some countries they cannot use the Etefone. So we decided to compare. These are 2 blocks. This is my flower cluster counting. In red, I have my UTC, okay? So we decided to compare early applications of Etefone, Etral, ACC, or NAA versus late sprays. And you can see here clearly that the early sprays are better. Often, anything that generates ethylene like Etefone, ACC would be better than NAA. We have seen that very clear. This is a proof of concept trial, okay? So the rates might be too high. We have to adjust that. NAA for Honeycrisp, if you go to 10 ppm, you're going to suppress size. If you have a variety that has problem with size and you spray 3 times, you're going to suppress size. I've done that. I've seen that very clear. Pears is even worse. Usually, Etefone is a better option, but you see clearly here that early sprays are even better. And here, even ACC, for instance, at bloom and petal fall, you have a little bit better here, but in this block, we had better return bloom the next year. Crop load, everything adjusted in that case. So these are just some take-home messages. I won't go through all of them, but I think we see that there is a difference in timing for flower initiation depending on the variety. I think early thinning is still so far the best strategy to ensure return bloom. And PGR summer sprays only work when the crop load is reduced early in the season. Because if you leave, let's say, you end up doing your hand thinning at 40, 35 millimeters fruit size, and then you load the trees with PGRs, you may have some increase in return bloom. But try to do your work earlier in this season. So this is what I have. I hope it helped a little bit. I'll be happy to answer any questions you guys may have. Thank you.